Unbelievable, but true. Scientists have discovered a way to make peacock feathers emit laser beams. You might be wondering, why on earth would anyone want to do that? Well, get ready for a fascinating journey into the world of science and nature. Peacock feathers, renowned for their vibrant iridescence, have revealed a hidden talent. A recent study shows that these feathers can produce laser light when treated with a common dye and exposed to a specific type of light pulse. The result? Two distinct laser beams, with wavelengths of 574 and 583 nanometers, emerge from the feather's eye spot. But here's where it gets controversial. The team behind this discovery believes that the laser effect arises from tiny structures within the feather itself, acting as a resonator. This challenges the conventional understanding of lasers, which typically require mirror-based cavities. The author suggests that something within the feather's intricate design is responsible for this unique behavior.
For many technological advancements, nature often provides the blueprint, says Nathan J. Dawson, a researcher at Florida Polytechnic University. These findings open up exciting possibilities for exploring complex biological materials and developing new light sources that can safely interact with living tissue. The concept of a biolaser is not new, but the use of peacock feathers adds a fascinating twist. The gain material in this case is the dye, while the feedback structure seems to be inherent in the feather's nanostructures. The cycling process of wetting and drying likely aids in diffusing the dye and creating a unique environment for laser emission. The eye spot colors of peacock feathers are not just a result of pigments. Instead, they arise from a photonic crystal of melanin rods embedded in keratin, which reflects specific wavelengths of light. This structural heritage gives the feathers their intense color, but the laser action requires the additional step of dye infusion and cycling. Researchers trimmed peacock feathers and mounted the eye spot region on a base. They then soaked the feathers in a solution of rhodamine 6G, dried them, and repeated this process several times. The key to success was the multiple wet and dry cycles, which allowed the dye to penetrate the feather structure and create the necessary conditions for laser emission. The experiments revealed two sharp peaks in the yellow-orange region of the spectrum, indicating the presence of laser light. The team observed the same wavelengths from different colored areas of the eye spot, suggesting a consistent laser mode within the natural structure. The dye-infused barbules, when subjected to wet and dry cycling, consistently emitted laser light across multiple parts of the same feather and different feather samples, Dawson explains. This behavior is not typical of random systems and points to a highly ordered structure within the feather. The green region of the eye spot produced the strongest emission, which aligns with the dye's absorption and emission characteristics and the local structure of the feather. The thresholds for laser emission were measured at around 380 microjoules per square millimeter in the brown region and approximately 290 microjoules per square millimeter in the yellow region. This discovery differs from random lasers, which can occur in biological samples without a well-defined cavity. Random lasers rely on scattering paths in a disordered medium for feedback, resulting in a spectrum that is highly sensitive to small changes. In contrast, the peacock feather laser exhibits a consistent set of wavelengths, suggesting a more ordered and controlled process. Previous studies have demonstrated lasers from bird feathers, but the new peacock result takes it a step further. By mapping consistent laser modes within the natural structure, this research opens up new possibilities for understanding and utilizing biological materials. The authors suggest that the feedback mechanism may arise from small, repeated features within the feather, such as protein granules or dye nanocrystals. These hidden building blocks could be responsible for creating multiple low-quality resonators with similar optical lengths. Using laser emission as a probe offers a unique way to uncover subtle order within complex biological tissues. Stable spectral lines that repeat across a sample indicate the presence of recurring microstructures that may be challenging to identify through traditional imaging techniques alone.
This optical fingerprinting technique could revolutionize the characterization of biocompatible materials and lead to the development of sensors and imaging tools that work efficiently and safely within living systems. However, further research is needed to identify the exact structures responsible for this phenomenon and understand how processing steps can be optimized. The study, published in Scientific Reports, highlights the incredible potential of nature-inspired technology and the endless possibilities for scientific exploration. So, what do you think? Are you intrigued by the idea of laser-emitting peacock feathers? Share your thoughts and let's discuss the fascinating world of science and nature.